recording.
Wonderful. Okay. We'll also open chat so I can see you. Yeah, so we are already behind, even though not everybody is here. So I'm just gonna start. So I did gave this presentation a couple of times in the past, but I don't think I ever made such an practical version of it. It's truly like Multiomics like integration primer about like why do we want to integrate, what are the problems with integration, and then so hypothesis more than a practice about the single molecule sequencing and multi-omics integration. Right. And then if we have time, I will show you my model as an example. Okay, I cannot switch now, so that's
maybe try the other guy. Yeah.
All right. So I will try to make this all to poor victims that are in the class to talk with me. So basically we all know that biological data sets are complex, and in modern times they are even more complex than they used to be. In general, some of this is because of the technological development, of course, but some of this is also just because of logistics and like experimental design, right? So at this level, I estimate that probably none of you would have a problem if you got an expression matrix like one here. Not sure how I can point, so I will just hope that you know which one of those is gene expression matrix. So I don't think most of you would have a problem with figuring out what are the samples and what are the features on such a matrix. So in this particular case, these are either genes or transcripts. If you are measuring different omic, like ataxic, this could be like regions or positions in the genome in case of mutations or like methylation, right? Frequently, we will have more than one omic measured. So this creates this particular problem of usually you wouldn't have an ideal situation of having the data point for each omic in each sample. Frequently, this will be because of like just technical troubles like dropout, but also like logistical problems. So you have a bunch of patients and maybe patients dropped out from the study, or they are not, not following up later on. There's not enough sample to measure all of the omics, etc. So that's like one of the ways that the missingness is represented in multi-omic integration. The other thing is obviously if like Chris already told you about the number of genes and number of transcripts, the like 20,000 or 60,000 human genes or protein coding genes or transcripts, it is like high rank data set, but it's still relatively small. If you try to analyze the methylation data set or a whole, base by base whole human genome, this would be big enough. Yes, over 60,000. Sorry, I am behind.
Total genes. Yes.
So this would already be like large scale data set. And if we introduce like additional omics to the mix, this becomes like bigger and bigger. So the cool fact that I think I heard from Jan Krovzik, some of you may know this, is that why do we usually, like in terms of gene expression and many different omics, have genes in like rows and not in columns? Do you know? It's a tradition.
Excel, I think, right?
Yes. So in the past, like there were many problems with people opening the datasets in Excel, and Excel has a maximum number of columns of like 16,384, and then it would just drop out. Did it even tell you? Just drop out the rest of the columns, which would definitely, like some of these papers were published before it was identified as an issue. So this is how it is. I know that there are different omics in which like the tradition is to put them in the other way, like metabolomics is one of them. but generally you would have like many and many rows. The fun fact is that if you try to analyze methylation, like 500K, that's like the most frequently used human methylation array, microarray, you would use, I think, like 14 gigabytes of memory in R to like just open. R generally, like if you open it normally, it would use twice the amount of memory it requires to later hold it in the memory because it's like not optimized this way. So this is definitely an issue. The other problem is, again, logistical. So frequently you would have different time points, you would have different sites, etc. So here we have like microbiome abundance over different time points, right? So there's additional level of difficulty. Like we already talked about how like the different different additions to RNA to the DNA to RNA to protein dogma appeared over years. There is definitely a lot of technological advances that appeared just in recent years, and specifically here I'm talking about single cell sequencing, which is, I guess, the thing of the last 10 years, if I had to select one. So we can profile definitely different omics in at the level of cells in the same cells. And we can definitely see like DNA sequence, cell surface proteins, epigenetic features, etc., at the same cells. This gives us an insight into heterogeneous nature of cells, and we can, probe the regulatory maps through like multiple omics or layers. Of them are also called modalities. There is not that much difference. So rather than looking at different modalities separately and then correlating the downstream analysis, we are trying to, here we're talking about integration. So we're trying to build a mathematical model to integrate and jointly analyze data, right? So why would we do that? Basically, organism is complex, not only data sets, but also organisms. So this gives us systems levels into etiology of disease that are useful in like biomarker prediction, diagnostics, driver genes discovery, etc. They might not be possible to be discovered when you analyze them separately, because, for example, it's not like a linear correlation between features in one omic and the other omic, right? One of the things that are not mentioned here in this complexity is that by nature, biological data sets are not really in the same like time scale, right? So like you have the feedback loop of expression, and then change, the changing DNA, so mutation, changing expression, the amount of product that is impacting the expression, and the other way around as well. So you may see the change at one level that is not yet available at the other level, but is coming, right? But in the different time scale. Right. So I mentioned that we can talk about multi-omics, and we can talk about modalities. As multimodalities specifically appear more frequently when we talk about measuring something at the same samples, at the same granularity, so like at the same cells. So here is a relatively recent comparison of technologies that allow to measure different omics at the same granularity. These are simultaneous assays that more or less recently emerged. You can look at the dates here, or they're all relatively recent. And some of them are in the same cell, some of them are different omics like independently, which means that they produce like unpaired data. Frequently, I think like mostly, this is like two omics top. Sometimes they're free, maybe. Dogmatic is a good paper to look into if you want to look at the integration of more than two omics at the single cell level. And right, so if we have data sets that are unpaired, what we are talking about in integration is so-called diagonal integration. This is the common experimental setup would be to just get like a tissue homogenized, and then the assumption is that the separate cell types will distribute evenly amongst different like aliquots, and then the divided sample will be run on like different assays. That's how you usually do like unpaired multiomics sample preparation. Of course, there are also public datasets, and the most notably, if we have time to go through my model, this is model for bulk datasets. So if you use like big biobank data, like TCGA, UK Biobank, etc., what you would have is the samples that might not come from the same sample, but they would come from the same patients, right? So then it is very important to think about where your sample is coming from and what is the experimental design, because this limits what models you can use in integration and like basically what questions you can ask, right? So if you use the exactly same sample, exactly same tissue to split for different omics, you can ask about mechanisms of how things are happening at the cellular level, for example. If you look at the patients and like a big cohort of patients, obviously you are population level changes, and these are all despite how much single cell data is produced right now, these are also very helpful, and especially it's good if there are public data sets. In my opinion, it's good to start there and like try to see like overview of what you can do, and then try to analyze or generate your own data. All right, yeah, so here the blue ones are the technologies that measure multiple modalities one cell at a time, either in individual test tubes or in microwells or in plates. So this is limited by the throughput and relatively high cost of each assay. And brown methods are process thousands to millions of cells, mostly using like droplet technology. Or combinatorial like DNA barcode strategies. So my question is, yeah, so I have so much problems that we already talk about. So like the different time points, different types of samples, different levels of information. So what is the biggest challenge of multi-omic data integration in your opinion? All ideas are good.
I think one is that there's just like no standardization of pipelines for a lot of omics in general, like even within their own omics. So how do you integrate something where you don't really have like standardization?
Can you hear class on Zoom? Just checking in.
Yeah, we can hear. Yep.
Yeah. So you talk about experimental, or do you talk about analysis?
It's both.
Yeah. So yes, that's true. Pipeline wise, I am as you know, I am less on the side of the wet lab. So it's hard for me to even imagine like all the intricacies that are going into the like design of an experiment that has multiple modalities from the same cells even. There is something that is also like basically a combination of what you're talking about, the standardization of like lab part and analysis part, which is batch effect, right? So batch effect, we will talk about this in a second, is something that is probably the biggest downfall of multi-omic integration usually. So this is definitely a thing. All right, so what else?
I think from like, this is further downstream, but I think that the goal of integration, right, ends up being something along the lines of causal inference. Can we say that changes or alterations in one data set predict changes?
Right.
And that's,
Correlation does not mean
worried about the batch effects and everything like that. It's really difficult to get at that.
Yeah, that's very good insight. Not exactly what I guess I meant, like more basic wise, but that's a very good insight. Yeah, this all goes down to like you need to think about the experimental design and what is essentially possible. And in most cases, what we're talking about here is we will integrate the data set to find the correlations, and then we will identify the most likely ones, and then we will do like downstream analysis to like confirm the mechanisms. That's usually how it goes. Yeah, so what is the problem with let's say integrating the gene expression and somatic mutation data, for example?
I have ideas, but the class should answer yes.
Actually, I've been trying to do that for one of my data sets, and one of the interesting things is that at least mutational data, like there's not necessarily directionality implied. So you have kind of incompatible types of data.
Yes, so data heterogeneity is a problem.
Yes.
Yes, different data distributions, like the of underlying distributions of different omics, definitely. Something that I specifically mention the somatic mutations, because you may think about somatic mutations as like binary data, right? You either have mutation or you don't. When you try to integrate binary data set with like continuous data set, what will immediately happen is that the binary data set is going to drive all of the distributions, and then continuous is going to be like went on like a second, you know, like everything that is correlated maybe with some mutations. And yeah, this is the problem, right? So different types of data, and especially sparse data sets like somatic mutations are specifically very hard to integrate, and you have to think about it when you create your model. And I have examples of like how it can be done, so we can talk about it if we have to. The other way of dealing with this like different distributions of data and data heterogeneity would be like performing like feature selection. Maybe if you have like large data sets and you have features that you think might not contribute that much more, you could do feature selection. There are obviously downsides of doing feature selection, because you by hand, like per knowledge, limit the dimensionality, and even features that are usually it is done like per variance, for example. Even features that are you know, not very variable can potentially have a combinatorial effect to like actually like separate, for example, clusters of patients between each other. So that's the problem if you have to feature selection. But again, the curse of dimensionality, like we have so much data, sometimes we are not even able to like model something so large. Maybe we need all of these GPUs in our Google Colab, for example, right? So this is one of the important things as well. Other thing is, like we already talked, that the different number of samples are per data type, but there's also different number of features, right? So like if you don't perform the same feature selection on all of the omics, then again, like one data set may like just decimate the other, right? Like in our downstream analysis. Right, and then from the differences in pipelines in wet lab and dry lab also comes the fact that, well, how do you deal with it? Normalization, right? So like the normalization across data types will be following different distributions is problematic, right? How do you create something that like contributes to the model in similar way? I think like truly batch effect is something that is frequently overlooked, because people say, oh yeah, we know that there is batch, and we try to avoid batch effect when we generate the data, but not frequently you have access, like you're able to be a part of decision as like computational biologist of the experiment being designed. And yeah, how do we deal with this? So if we just like integrate data sets like ad hoc without keeping track of like any metadata, this is what we'll probably see. So yeah, so batch effect. Batch effect is non-biological variation. It's come from like technical side. So can you give me like an example of why there may be like batch effect appearing in the data set?
Different measurements at different times, different locations, different
Yeah,
people.
So different.
handling samples.
Yeah, even the same person, but like one morning they had coffee and the other they didn't, right? Like, or you have so much data, especially in multi-omic like experiments, that it's frequently not possible to process them at the same time, right? So like the natural degradation will also happen in datasets, specifically if you use like RNA, right? That is like easier degradable, so it definitely can happen. So there are also, this basically makes the samples not directly comparable if we have some technical variation. So let's say we have like cancer and normal, right? So normally, like if you would have a cancer and normal sample from the same patients, how it would happen is you would extract like neighboring tissue to like the tumor that you're extracting. Like on the way to tumor, basically, to have like a control, right? PERT control. So let's say we have like multiple samples, same cancer type. How do you escape samples like clustering per, patient and not per like condition? You basically have to like look at your experimental design and regress it. That's the way. That's the term that I would use right now. But maybe we see the same samples clustering there for a reason, right? So all of the regression will assume that this is the technical noise that we want to remove, right? So yeah, so like I said, different mice, different sequencers, like protocols, etc., right? So these are all potential problems. In big biobanks, you would have very meticulous, hopefully you would have very meticulous metadata, and this is the way to like figure out if there were changes in the design. So how do we avoid interference into data that would help us remove the batch effect, but will also like mess up our biological significance. So here's my favorite example of batch effect. This is actual paper that was released in 2014, and by I think these were like ENCODE people, if I'm not mistaken.
Yes, this is the classic. Yes.
Yeah. So right. So in this particular figure, did any of you use like model organisms in your research before? What did you use? Mice or something else?
Yeah, like C57 black 6 mice.
Yeah. Yeah. So this very cool example just shows us that basically like the all of the human brains and all of the human like heart, for example, all of the human tissues will be more similar to each other than their like model organism equivalents, which makes all of our model organism projects and contributions basically useless, right? Of course, like sometime later, people like discovered this and actually did a batch correction. And I don't know, you cannot see this well because it's very small, but if you just introduce the labels from size that the tissues were sequenced in, you see that the distribution of like sample similarities changes greatly. So first of all, yes, good experimental design and like keeping track of metadata is probably the most important. In terms of good experimental design, what is important, I think, is that you avoid like perfect confounding. Perfect confounding means that groups are perfectly correlated. So like, for example, you have treatment in batch 1 and control or treatment 2 in batch 2, right? So like then you cannot really distinguish clinical versus biological difference. The best way to go about it would be, if you can impact the experiment design, is to balance the groups of interest. So include at least some control samples in each batch. And if you can include the same control samples split into like what, that's like perfect way to go about it. But if you suspect that you have some batch effect and you want to remove it, there's no way, then iterative way. So basically what you would do is, and I think that's the most important point of this talk, is you take a look at your data. That's you always take a look at your data first, and then you identify that you might have a problem. Then you try to correct it, and then you visualize again and try to see if there's still an issue, or maybe you overcorrect it, right? Like this is all. It feels more like an art than science, but that's how it works, right? And usually, like I would recommend Combat, like classic 2007 paper that uses like empirical Bayesian adjustment and was originally for microarray data, but can be used on different data types to correct the batch effect. There is also, data type specific. So lemma here as an example, because it's obviously a package for RNA-seq, like RNA expression analysis. This particular one, though, implements the complex, like nested batches. So we might have like different sites, like different hospitals, and then inside we might have different people that run experiment, for example, right? So like this is the nested batch effect. Yeah. So, or like, maybe you have fresh and frozen sample or whatever. This is like additional level of like nested batch. Right. So yeah, so this is paper. This is you can see that this paper is from 2007 by the resolution, but yeah, this is what you do, like iteratively, directly modify your data in an attempt to eliminate the batch effect. Something that I would recommend is, especially if you're doing multi-omic operations mostly, is the unsupervised like exploration of data. So you would probably cluster, and then once you cluster and once you start your downstream batches again, because maybe you need a bit more correction, right? Yeah, I have a question.
Yeah, so I did some work where I was trying to integrate some mouse data with like the TCGA, like expression data. And one thing that I thought was really interesting is that for their data, it didn't have, like a ton of batch corrects or like batch effects internally, because they had a very standardized pipeline for sequencing and analyzing and analyzing their data. But what's interesting is that adding only a few of the mouse samples to that actually resulted in the mice samples clustering like with the tumor types that they were supposed to cluster with.
Yeah.
And I always wondered whether if you have, like a robust cluster, if you have a huge data set and you just want to integrate another data set that's maybe smaller, that will have batch effects because it's processed completely differently. I always wondered if you just did the clustering with only with each sample from your data set one at a time with the original one, whether you would actually get correct clustering.
So that you didn't see any difference was probably due to power, like basically like the biological effect was much bigger than the technical effect. There'd still be some some impact, like moved samples in like other direction a bit, right? Like because of the technical power. The batch effect is not always going to be there, right? I'm just saying like be careful, because if there is something weird in the data, like 9 out of 10, it's batch effect. They are like semi-supervised models, and the semi-supervised models literally do what you're talking about. So you have like you basically create a distribution of your data based on samples that are more sure of, like more fuller, like less dropout, for example, samples, or samples that have 3 omics, and then for the rest you have just 2, right?
Yeah.
Then you used the model to basically assign the samples to different clusters. Most of them actually, like my model can be used like that. The only difference is basically you minimalize different things. So you add to your objective function to minimalize the differences between like label, like the samples, like the clustering is the same, just the samples with the same label should be in the same cluster. So you add additional constraint. So this would like also increase the power to like be able to move the samples to actual cluster where they should be. but again, there is no, it's art, right? Like there is no just one rule that will work for everything, but yeah.
But do you think it's even possible, like, I guess what I originally, when I interpreted that data, what I thought was happening was that it was impossible for the clustering algorithm to even learn what the batch effects are, because I was only adding a few samples at a time, so it didn't capture that as a major component.
Uneven labels maybe. I think like for mice and human, you probably like you had to map, like there's a bunch of steps that you had to do, like and each of them like map mouse genes to like human genes. There was probably some filtering, there was like normalization data and general data. Like there's a bunch of steps that, you know, were together contributed as in an integrative way to what happened. Yeah. It's hard to tell.
Yeah.
That is very specific to like single cell and potentially very interesting for the single molecule sequencing is that the frequently we would just have like completely distinct like feature and sample space. So this is where you have like, you know, the same sample maybe, but the different cells are coming to like different omic assay, right? So like there are different ways of dealing with it. One of them would be like matching cells that have like similar profiles, and this is usually like some non-linear fold alignments. Which this removes like a prior knowledge completely, but it does requires a lot of assumptions, right? So like, or maybe you have some cells that are actually like mapped in both cases, right? So that can be used as a control. And something like that was like mostly applied to relatively small data sets with like limited number of cells, because like computational power as well of cell matching and many assumptions. The like explicit feature conversion is also an option. So like you convert multimodal data into like one common feature space based on your prior knowledge. Then you apply like single omic data integration methods. This is straightforward. Again, major information loss. And if you have a lot of dropout in one omic versus the other, you come to the same issue of like uneven feature contribution to the feature space. There is, and something that in most cases will happen is that you will basically do a joint matrix factorization, which requires no explicit feature conversion, but is computationally less scalable. So essentially, what happens is we try to find like low rank representation of our data. Like in general, in bulk integration methods, what you would expect would be like much more features than samples. And you probably saw this plot many times. Probably there is an updated version. This is from 2016. So we have data sets that routinely run like in the scale of like millions of cells, right? So so the methods of integration should be designed with like scalability in mind, which means that some of the bulk methods technically are applicable to single cell, but they will just take much more computational power, right? This is something to keep in mind. So generally, what would happen is in representation. Okay. So what would happen in almost all of the cases is you just try to find integrative low-rank representation of your data set to go from like data representation that is full to reduced. I didn't want to go into a lot of linear algebra, but basically the slide, it encompasses whatever you want to, like PCA, single value decomposition, matrix factorization, etc. They all, like most of them, work in this way, which means that you build a model, like you assume this is my distribution at the beginning, like this is my matrix, and then I want to get like a low rank matrix, for example, and then this low rank matrix, I put constraint on it. This is done by an objective over cost function. So for example, I want this low rank matrix to be sparse. So which means that I want samples to be more confidently say that they are in this column and not the other column, which frequently means this cluster and not that cluster, right? So this is the sparsity component, which is very frequent. If you add semi-supervised, like we just discussed, this is where you would add in the cost function differences between labels. So usually when you build like your objective functions, what would happen is that from how you arrive from left to right, it's iteratively usually some like gradient descent method or something like that. The easiest way to go about it would be just, start from random, update the matrix, see if the result is like close enough. If not, update it again, see if it's close enough, etc. That's how most of the gradient-based methods work, or iterative updative methods. If you want to go into the algebra of it all, I would recommend this particular book. So for multi-omic analysis, because it will give you the algebra, but it will give you explanation in the bioinformatics sense. I think it's pretty good. But yeah, maybe we'll be back to the math in a second. So what happens if this is even more complicated? So we have many data sets. Well, there are different approaches to multi-omic integration. So this plot is a bit dated now because it's from 2018, but I think it holds up pretty well. So the easiest way to integrate, again, would be just same feature space, right? So we take our, we have an integration, we just concatenate omics, and then we just cluster, for example, right, to find our samples. Yeah, the problem with it is unequal feature contributions, different data distribution, etc. The method in red is the method that if you did any multi-omic integration or clustering, you probably see this one. These are like well-established methods for the examples. The statistical models, they require usually like a probabilistic modeling. And this is where the distributions of data sets can be like explicitly, distributions of different types of data can be explicitly given. But the problem is that they would frequently just be limited to the data sets that you, like, they wouldn't be generalizable, right? Because you assume the distributions, right? And sometimes they are heavy, unless they are like Bayesian versions. The late integration, as opposed to early integration, that would be basically analyzing the data sets separately and then attempting to integrate at the level of clusters, which is how, am I gonna be in trouble if I put this opinion? This is how most of the WHO, updated molecular classifications of cancers were probably derived, at least the ones that I know about. So frequently they always say it's a multi-omic integration, but they would probably look at what distinguished the samples the most, like maybe specific somatic mutation, and then like random forest, like, right? Like they would like look at the inside the clusters that they created with different omic, for example. It's not perfect, right? Because the combinatorial power of the omics to discover the potential is limited if you analyze the dataset this way. But it's also like pretty easy to validate, which is why it's used. At least in the well-established, older classifications. And then we have this two ways of, so one of them is, we already discussed, dimensionality reduction methods, whichever you would like to choose. And then the other would be like similarity-based methods. So you look at each omic and you try to find which samples are the most similar to each other. And then you try to integrate the similarities. And then this is my program, Sumo, who is using actually both of those to kind of jump over and try to create like generalizable methods that does not like makes any many assumptions about like the distribution of the data, right? So what time is it? I should probably be finishing, but our second speaker there has
Rob,
clinical.
Rob is coming from clinic, so I guess you can go until he gets there. Yeah.
Cool, yeah. So I'm just gonna jump a bit for this part. So again, so this is Sumo. This is my model, but again, like preface is this was created for bulk data, so it does assume that you generally would have like more features than samples. But I think like I mean I am very happy with how generalizable it is. The fun part is that the generalization wasn't the most important part. What was the most important part is the missing data. So surprising amount of models that you use actually requires imputation of the data. So you have a lot of drops, right? And you need to like make your matrices complete without missing values. And you can do it by dropping the features, or you can do it by basically simulating the values that would be there. Frequently, this is done based on nearest neighbors, right? So you look at the gene and you look at the similar genes, and like so the samples that have like similar expression, like distribution, for example, and then you simulate the missing gene value for a sample. And in this particular model, the way it works is it uses matrix factorization. And this is non-negative matrix factorization. So here is the algebra, but it's gonna be quick and it's gonna be easy. So what we are starting with, let's imagine that we have our like gene expression matrix on the left side, and then we want to find our low rank representation. That would be the W here matrix, right? On the other side. So again, what we would do is we would try to like, so we have the left and we want to do the right. So we just do a random values on the right side, right? And then we check the difference between the left side and the right side in the values. So right side is the factorization of two matrices. If you factorize them, you will have exactly the same, size of the matrix on the right side and the left side. So we can just pairwise compare the values, or if you can do it using an actual mathematical metric, which truly does the same. It does connect the values, right? So like this is when you do machine learning. This is where you learn about like L1, L2, etc. That's the level. Essentially, let's say we start with random. Then we're going to update one value and check if the difference between left and right side is bigger or smaller. If it's smaller, then we are closer to our perfect solution, right? So we can then start from this updated version, and then we can do another change, right? And then this is how we arrive at some point to the almost good enough, because usually it's not going to be perfect, to the like error margin of the distribution, and this is our low rank representation, right? So in this case, this was done originally on the feature matrices. Now, how about we do it on similarity matrices? So as long as we figure out what is a good metric for our specific omic to figure out like sample similarities, we can create a similarity matrix, which was going to be square, and it's going to be non-negative, which is the part for the math part here, right? So from 0 to 1. And then this basically we go over the distribution problem. We just sidestep it, right? Because from this point, we're just going to work on the similarity matrices. Does it make sense? Okay. I see some notes in the class. Hopefully on chat is the same. So, yes?
Yes.
Okay, yeah. So so we have three matrices. So this is like 3 factorization technically, but then this one matrix here, this is just a specific version of non-negative matrixization called symmetric non-negative matrixization. So this is the same. So now this is done for each similarity matrix. So let's say we have our expression data, and then we have our methylation data, right? So we have two similarity matrices. This H matrix that we want to get. We want it to be the same for all of the samples because this is our joint low rank representation. So yeah, so this is our problem on top here. And then I don't know if you can see my mouse on the Zoom, hopefully. And this is our cost function, which again, I know math, but essentially here's what we're doing, right? So we are checking for the difference between left and right side. And this is Frobenius norm, which like essentially like checks kind of element wise and just sums it up. So we can think about it. Now, to support missing values, here we introduce like 0-1 matrix that will literally just remove through our sum some values that are missing. And then I also weighted around to like take into account that different omics are going to have different numbers of samples. So in this case, that would be patients, right? For which we don't have available data. And then here we impose what we want. So we want this new low rank representation to be sparse. So we want the clusters to be moved as far as possible across different like axes, right, from each other. So we want to be sure, want ambiguity. And yeah, and this is literally like a gradient, like you may imagine, right? So like, if you want to talk more about math, I think I'm gonna stop at this moment, but I could go into detail separately. And then we incorporate a bunch of different tricks. Yeah, a bunch of different tricks that makes it more stable and make sure that we don't do it just once. We use some like resampling based approaches to make sure that we are like to increase the increase the chance that our results are stable, basically. And I think I'm gonna jump over this unless you're interested. And I'm just gonna talk about some biology. So this was benchmark on TCGA, and generally it did like that pretty well. It worked for all of the methods that different samples work, and then for some samples, like this ovarian cancer, for example, it identified the groups of patients that had either the significant differences in survival or significant differences in clinical like labels enrichment, like age, for example, or sex. And then like we focused on the LGGs. So here is the integration of the expression, DNA methylation and miRNA expression. Briefly, for low-grade gliomas, this is like 5% of all primary brain tumors. This classification includes oligodendrogliomas and astrocytomas. They're associated with like wide range of symptoms from like seizures, cognitive impairment, etc. The WHO classifies the low-grade gliomas from grades of 1 to 4. And usually, if you would go to the doctor, and you would have like a histopathological sample taken, so biopsy, and this is how like they look at the sample and they check, for example, for necrosis of cells and like different this type of features. And then this updated 2016 classification includes also like molecular factors. So you would also have like a gene targeted array done. And these are the mutations that would be used to like distinguish different types of cancer. Right, so basically molecular setup of some genes and some regions. So when we used Sumo to integrate these data sets, what we noticed was that we have, since Sumo uses kind of like a greedy approach, because there's no good way to select the number of clusters for non-negative matrix factorization, we included like some of the methods that we think are, one of them is very specific. This is a new method and new metric that is specific to this method because we use consensus clustering. So we basically do clustering many times, and then we look at like consensus of how many times two samples were together in one cluster. And then this is how we calculate the proportion of like ambiguous clustering. And by ambiguous, I truly mean like between 10% to 80% like of ambiguity. They should be never together or always together. That's the perfect case. And then this is more like non-negative matrix factorization specific method, which is cognitive correlation coefficient. So essentially what we checked was that we have two like stable solutions for 2 and for 5 clusters. two clusters. This like recapitulated pretty well what was seen in the in this separation, specifically the like majority of ADH mutants with either this, like I will point it here, 1p 19q codeletion, right? So and with higher global levels of DNA methylation. This is the cluster with the better prognosis on the middle panel. But then we noticed that there is this very, very interesting result that showed that in subtype 2 here, so the blue line, includes a lot of samples that would be classified like as a grade 2-3. So so they wouldn't be treated, you know, as like maximally malignant, right? And but they display a molecular similarity actually with like grade 4 or even glioblastoma, like the and based on histopathology, they are just grade 2-3. So what happened here? You know, we checked it, we did like integration multiple times, adjusting different parameters of the model, and we still had this cluster of samples that normally these patients would grade 2. And overall, all of the samples grade 2-3 had pretty good survival, but there were just a bunch of patients that were separating and had terrible survival. So based on downstream analysis, we essentially all of the data omics that were available, but they weren't used to integration. We used evaluation of enrichment. Never calculate p-values of enrichment based on the omics that you actually use for integration, because that's just the assumptions of the statistical tests are not met. So here, what we checked is that this cluster 2, right? This is the blue one, actually has a pretty good enrichment of chromosome 7 gain, chromosome 10 loss, which is actually correlating with this, particularly these two, but many mutations that are like point mutations in gliomas in generally. So particularly like EGFR, this is the epidermal growth factor receptor, is an oncogene on chromosome 7, right? So if you have gain of chromosome 7, you have a gain of copy of this oncogene. Which, you know, amplification and like division of the cells is impacted. Then we have this P10 is a known tumor suppressor, and it's on chromosome 10, so we have a loss of tumor suppressor. And generally, this cancers, these tumors would like develop much faster, and even though in histopathology you wouldn't see it, you should probably test patients for it. And just give them more aggressive course of treatment to like, you know, make their chances better. There were additional changes here, generally like genome-wide hypomethylation and chromosome 7 gain and chromosome 10 loss appear to be the hallmarks of this type. And we published this this paper calling for the change of the WHO criteria, or like inclusion of this particular one. There is a couple, this is not fully novel result, right? Because the pathologists, there's a consortium of pathologists called C-Impact that based on their own cases, like they already seen that if they did like more wide panel, you see like whole chromosome 7 gain, chromosome 10 loss, or like one, you know, one arm of the chromosome. But this is like this is where the multi-omic integration came to like validate this, right? Integratively. Yeah, so either just including this to create like a subtype of a subtype, or just creating like additional subtype, which would be low-grade glioma subtype with molecular features of like glioblastoma. Yeah, and like this, here's some additional validations. You know, this is bulk data sets. Tumors are complex like milieu of different cell types. So, so obviously the hypothesis is that the microenvironment plays a very important role in the determination of the subtypes. So here, if you are using TCGA samples, there's something called X cell, not XL, X and then cell scores, which corresponds to enrichment of different immune and stromal cell types. And these are derived from like gene expression, but they are actually like methylation equivalents, and they are, I think, derived with the tool called Mira. And yeah, based on this, you can see that this chromosome 2 and like glioblastomas are much more like similar than different groups of low-grade glioma samples in terms of like cell type composition. And yeah, and we look here and we see like there's like sure enrichment, of several cellular populations, but like astrocytomas are mostly assigned here in comparison to other subtypes. And here in the chromosome 3, we have much more like neurons. Right. So yeah. So, and there is a bunch of different like downstream analysis that we can do here. So one of the ones that I did was to look at the correlation of like methylation markers with the expression using a package called Elmer. And then thanks to this, you could like basically identify the like hypomethylated or hypermethylated regions of the genome that correlate with your samples, right? So for your samples, and this is how we like figure out that there's also like a gain of area response transcription factor complex AP1 occupancy in like demethylated distal enhancers, which is very simple. It's happening in glioblastoma.
But the regulation gets disrupted, yeah.
Hi.
I wasn't sure how long. I didn't want to interrupt.
Oh, you see, and I'm talking because you're not here. So we are like completely
Standing in the back, so.
yeah. So so of course, like come in. Like so I'm just going to conclude basically. How are we looking at the single molecule multi-omics level? Well, we already talked in depth in this studying transcript and like how direct RNA and single molecular sequencing is very helpful in this. There's not that much in terms of multi-omic integration of single molecule sequencing yet. We may hypothesize about how it would look like. Essentially, like we are meeting the same issue as we discussed with the single cell models. So we have completely like distinct features and sample space. But this depends on the design of our experiment, right? So if we're using transcripts as features versus transcripts as samples, right? So yeah, so there are methods that allow us to check base modifications, and we can relate base modifications with like transcript expression. This definitely we can check multiple base modifications, and usually what we would have would be then like k-mers across transcripts, and then like probability of one modifications versus the other, right? So this adds additional complexity, and obviously complexity that's not taken into account here at all is the single structure of the RNA that is impacted by the base modifications. So which makes this field of like single molecular potential multi-omics very exciting, but also like truly this is yet to come. It's a matter of time. And right now, from my experience, like the RNA secondary structure formation is frequently just used to you use the it for validation when you do your like base call or base modification, and then you either identify different structural enrichment or you validate using known structural enrichments. Yeah, and then please look at your data. Very basic things. We talked about batch effect. So like make sure that you know what is the experimental design, and like just keep track, just write it down if your metadata, because it may come helpful at the end. And each model has an assumption, and this is very important to check. And if you are not sure, you can always try to simulate the data that you know the answer to, or and double check. Yeah, single molecular omics are on the way. Right, that's this part. some acknowledgements, but this is mostly for that funded and worked with me on the Sumo part and my current affiliation. Do you want to join through your Zoom?
Thank you. We should have, yeah, Dr. Schwartzstrom. And yeah, say metadata is a love note to the future. I will say there as well. Great. That was originally from Jason Scott. So actually I might even, I'll just close this recording and I'll start a new one for the new lecture and
Great.